It says we're live. It's been right the past few times, so maybe we should believe it this time. Okay, dear audience, welcome to another edition of It's the Science Bitches. I've done a JD Kane again.
I was just about to type that.
Yeah, I had it turned. I thought I turned it off, but I just turned it down. Damn it. Anyway. Your hosts, the Science Bitches, me and Jade. We are the Science Bitches. We're bringing you science. We also do have two of our honorary bitches with us. We have our RSC, known to his friends as RC, and Cliff Jumper, the tech guy.
Woohoo.
For some reason, two have gone missing. A sort of AWOL. They have the link. If you are watching this, the pair of you, you'll find the link in your G+. But you are missing, and it has been noticed. Our other two honorary bitches. But meanwhile, me and Jade, we're going to do our usual thing. We're going to sit here and let our honorary bitch, RSC, do the hard work. He's prepared some slides. Now, this week we're talking about the immune system. What is the immune system? What's it for? How does it do it? And possibly we will get to what happens when it decides to no longer play along, or if you don't have one in the first place. But we'll start with the very basics of the immune system. And I'm only going to give a really brief introduction. For anybody who's interested in the topic, you can make a very good start by just going to Wikipedia and typing in immune system. And you'll get a really good overview of it there. And also you'll get the links at the bottom that you can then chase up yourself if you want to know more. I'll just start by saying the immune system can be broadly split into two different parts. There's innate immunity, and then there's the active immunity. The active immunity is the type of immunity you would get from a vaccine or from having had the disease, and it's specific. Innate immunity is non-specific in nature, and in fact, one of the biggest parts of innate immunity is the skin. A lot of people don't actually realise that. But then if you sit back and think about it, look what happens to burns victims. Without that barrier, a non-specific barrier, that stops anything unless it's breached. So it's stopping anything. It is not specific to a given pathogen. And without that barrier, you are basically a veritable smorgasbord of nutrients for various bacteria, fungi, viruses. The skin is is a really good example of the innate immune system. Now, what I am going to do is hand you over into the tender hands of RC, and with, I was going to say collaboration from Cliff Jumper, but we'll go with conspiracy from Cliff Jumper. He's actually going to do a little presentation here. Anybody who's got any questions, please type them in the live chat, and we will do our very, very bestest to answer them. And if we can't answer them, we'll tell you honestly, we don't know. We won't try and baffle you with bullshit. So ask away. Meanwhile, RSC, darling, are you ready?
I'm ready. Can you hear me?
Yes, we can hear you loud and clear. Off you go.
All right. Well, Cliff, thank you so much for agreeing to host and present the slides. That is actually probably more important than what I'm going to be saying here, because people are going to no idea what we're talking about unless they can actually see something. So today we're going to talk about immunology, which is the body's defense system against microorganisms, pathogens, and parasites. Let's jump to the next slide. Ooh, I don't know. Can people see all the information we have? So basically, we're going to start with a lot of definitions. And I apologize because immunology is basically a foreign language to everyone except for immunologists. But when we use the word immunity, we're basically talking about the body's system that defends against infections, parasites, pathogens, like I kind of said. It's a complex system that consists of organs, tissues, and cells. And there are also non-cellular components like antibodies that you may have heard of, and other factors as well that we'll get to talk about. We'll start talking about specific terms. So when I mention antigen, I think we've discussed this in the past, but antigen just refers to any molecule that binds and causes an immune reaction. Very specifically, when we say immune reaction, we mean a type of cell called a T cell. That's the cell that does the reaction, that binds the antigen and creates the immune response. It's basically the transducer that says, hey, this particle, this molecule, I found it. The T cell is the one that recognizes whether that's part of your body or whether that's something foreign. So let's talk about the different types of cells. So we just mentioned T cell. Lymphocytes are a generic category that we call these immune cells. They're called lymphocytes because they come from the lymphoid tissues. So like you have lymph nodes and you have lymph conduction in the body. This is where these cells basically travel and make their way fighting infection and protecting your body. There are two major types of lymphocytes. We just nickname them B and T. So B cells are called B lymphocytes. T cells are called T lymphocytes. The B and the T come from their site of origin. B is bone. So B cells come from the bone. And T cells come from an organ called the thymus. It's a specific immune organ in the body. It's kind of located underneath your sternum. So if you tap your chest, right underneath where your throat goes to the chest bone, it's right underneath there. It's bigger when you're a kid, and after your T cells learn how to identify your body from the rest of the world, that organ slowly shrinks until the point where it becomes very hard to find when you're an adult. That's just a little FYI. B cells and T cells, they grow in their native home, which is either bone or the thymus, and they divide in response to infection. When these cells divide, they go through a process we just call maturing, where they go and they acquire all the things they need. So the T cells have to learn how to identify, what is part of your body versus what is foreign, and the B cells have to learn how to make the antibodies. That's their main job. So once they're mature enough to do those processes, that's when they get released into the body to actually fight infection.
Can I ask a quick question?
Yeah, go ahead.
Now I understand that in some the white blood cells are the ones that seek out invaders and attack them.
Yes.
Is this related?
Yes. So white blood cells are also an umbrella class that are included here. I think they're on the next slide. So we'll get to them in a second.
Okay.
I just got to explain a lot of these terms. I try to put them together in a system that made sense because I went from immunity to antigen. So I wanted to talk about the antigen cells, which are the lymphocytes. They're the ones that respond to the antigens more strongly and direct the other cells. So when we talk about lymphocytes, they're related to two types of immunity that your body uses. One system is called humoral immunity, which we will talk about much more in depth, but it's basically the adaptive immunity that your body has. It's related by antibodies. There's also cell-mediated immunity, and this is another type of immunity based on T cells. T cells will orient the other types of white blood cells to say, this is foreign, attack here. This is not foreign. Don't attack this. They're kind of like the signal people who direct traffic in a certain regard. The signals that T cells use are called cytokines. And these are basically just chemicals that they secrete out that basically give direction to the other cells. This is like how they talk and communicate with one another. And there's also a specific type of cytokine called an interleukin. So interleukin is a cytokine that the lymphocytes use to communicate specifically between each other. So hopefully we kept that as closely knit as possible. Are there any questions so far on any of the definitions or anything we've talked about? Fiona, Jay, jump in here if you want to say anything as well.
No, I'm quite happy so far.
All right, let's go to the next slide. And this will be, I think, the last slide with definitions. So bear with me for just a couple more minutes. Okay, so leukocytes. These are the white blood cells. They're separate from the lymphocytes. Leukocytes are called white blood cells because they mainly stay in the blood and the tissues, whereas the lymphocytes stay in the lymph nodes and the lymph system. So we're basically dividing these cells based on where they hang out or where they grow and mature. These leukocytes are also called white blood cells, as you mentioned before, Cliff. And these are the immune cells that go into the blood and tissue that kind of hang out there and protect. They're the defense line that's already in the body, waiting in case something comes and invades, in case you get an infection, a bacteria or a virus shows up. Another type of cell that is a very important and probably the largest part of the immune system are epithelial cells. Epithelial cells just refer to any cell that makes contact between your body and the outside environment. So there's basically two layers or two areas where this is important. Your skin is the largest organ in your body. That is the primary epithelial cell. It protects your body from exposure to basically everything around you. But then you have specialized epithelial cells that form your gut, because technically your gut is a hollow tube that makes a connection to the outside world, even though it's inside of your physical body. They're the second type of epithelial cells that we might talk about a little later. From here on out, there are some more terms we're going to have to learn, and these are just descriptive terms of what some classes of these white blood cells will do. These leukocytes. Some are called phagocytes as well. They're a subgroup of leukocytes, of white blood cells. And the word phagus means to engulf or to eat. So as you can tell, when you combine phago and cyte is cell, you get a cell that eats other microbes or particles or other cells. Dendritic cells are another type of leukocyte. Dendritic cells, they just basically present antigens. They capture antigens from the microenvironment around the cell and present it to the T cell. That way the T cell doesn't have to do multiple things. It just has to look at antigens. Dendritic cells are the cells that capture the antigens for the T cells. The next type of leukocyte we'll talk about is natural killer cells. And they're called natural killer cells, even though they're a subtype of T cell. But they basically will kill infected cells. If cells are infected by a virus, they'll start to display certain markers on their surface. And natural killer cells recognize these signals and will destroy the cell. They also work in a great regard to identifying tumors. When we've talked about in the past, tumors suppressing the immune system or tricking the immune system, we're mainly talking about them tricking the natural killer cells and their ability to destroy tumors. So antigen-presenting cells, this is just a large class of leukocytes that are mainly like the dendritic cells. They take up antigens from their environment. Basically, they digest them into smaller bites of antigens, and then they put them in their surface. They put them in their cell wall or their cell membrane. And that way, antigen-detecting cells like T cells can come, and they basically feel the surface of these antigen-presenting cells. And that's how they understand what type of antigens are present in the body. And then they can identify whether these are good antigens, these are part of the body, or these antigens are foreign. And so it's done all through this very intricate system called an MHC2 molecule. If you ever want to look at some of the intricacies of how this is actually done, look into Google, look in YouTube, and type in MHC2 class molecules, antigen presenting cells. You'll get some really great videos. And hopefully we'll have time at the end to look at some of these videos as well. Last two definitions, effector cells. These are just cells that eliminate microbes. This could be a lymphocyte. This could be a leukocyte. There are cell lines in both those categories that do this, eliminate microbes. And opsonization, this is the marking of a particle, a bacteria, or a virus for destruction. So the cells have to communicate to each other. They do so in many ways. We've talked about the interleukins and the cytokines as markers for communication. But opsonization is basically the universal signal to say, attack here. It's the big red X that you see on the military map that tells the army where to attack. Opsonization. So at this point, let's pause and see if anyone has a question in the live chat or if there's anything we can address.
Currently, the only questions that we have, I think you're actually going to address later on in your presentation.
Yeah.
But I think give you a heads up, to concentrate on it. Tommy wants to know, and what was that question again? Where is it? Let me find it again. How does a mindless entity such as a tumor trick cells? How do cells get tricked?
Well, it's really like an analogy that I'm using. It's not that the tumor is doing anything with intention. Basically, the tumor begins to stop doing some of the functions that I've talked about. We talked about antigen-presenting cells. Well, technically, all the cells in your body are presenting antigens. They all have markers on their surface that T cells will come up and rub up against to detect and determine whether these are body signals from yourself or these signals of something that's foreign in the environment. Once the tumor stops presenting any type of things that are foreign in the environment or anything that's indicative of the carcinogenesis, the transformation into a cancer, that's how they get around that natural killer cell. Those T cells can't detect that, hey, this cell has undergone tumorigenesis. It is now a tumor. It is now a foreign type of cell. And instead, they don't present that signal. So the natural killers are deceived or tricked in a sense. Does that explain it well enough?
With Tommy, who knows?
Okay. Well, I think everyone will understand and appreciate it more when we get to the videos, because it's very difficult to visualize these concepts. It needs to be seen and explained at the same time. And unfortunately, I can't do that just through a simple PowerPoint like this. Cliff, if you're ready, let's go on to the next slide.
Well, I just wanted to, can you guys hear me okay?
Yep.
Okay. I just wanted, you don't have to go back to the previous slide, but I just wanted to make a comment about the language used. A lot of times people find this language very confusing. And if you look it up, you will generally find out why the language is what it is. For example, leukocyte. A leukocyte, cyte means cell. Leuko comes from the Greek word for white. So white cell, white blood cell. Dendric, anything dendric or dendrotic comes from tree. And these cells at one point in their development, if I'm correct, RSC, have a tree shape or a branch shape. And so that's where they got the term dendritic, because that has to do with C. And
100% right. Mm-hmm.
Okay. And, phagocytes, again, phago is another word, again, cyte, again, meaning cell. Okay, so every time you see cyte, it means cell. Phago means to eat or to devour. Again, we're going from Greek. And this is why when you study science, you learn Greek. You learn a little bit of Greek language, Greek's suffixes and prefixes. It helps you understand. So if you run into the word phage or phage somewhere, you know what it means. It means something that's eating or killing or devouring. If you run into the word leuc, or as in leukocyte, it means white, and so on and so forth. Just a hint for people: if you have trouble with some of these terms, go ahead and look them up. You can find the etymology, and it may make more sense to you and may make it easier for you to remember them. That's all I wanted to say.
No, excellent point. Yeah, I kind of glossed over that, the Greek connection there, but thank you for bringing that up and explaining it in more detail. So I initially kind of briefly mentioned like what makes up the immune system. It starts with tissues and organs at the macroscopic scale, and these things include the thymus and the bone marrow, like I said. Bone marrow is where all your blood cells are produced. The B cells specifically will mature in the bone marrow, hence the B cell. The T cells will form in the bone marrow, but then they will travel to the thymus. Cliff, I don't know if you can point to where the thymus is. The little blue area right up underneath the neck. Yeah, exactly. That's where your thymus is. And again, it's much larger when you're a child because that's when your T cells are learning how to identify what's you versus what's not you. And it because it is less used as you age, it shrinks and it's not as prominent. Other organs.
Question: as prominent, but also as effective.
Well, the thymus doesn't really have an effect. It's basically just a housing area where the T cells learn how to identify self versus non-self. Once they've learned theoretically how to identify self, they don't need to go to school anymore in the thymus. They don't have to live in the thymus. They can just have a small population that stays in the thymus, and then when there's something that causes an infection or an illness, they travel from the thymus to the site of the infection, and then they divide there and basically get reinforcements by growing and dividing closer to the infection rather than growing and dividing far away from the infection in the thymus.
So to use an analogy, they don't need to go to school. The school doesn't necessarily shut down, but it doesn't need to go because it only needs to go through refresher courses.
Yeah, that's a nice analogy. Basically, the population at the school is not as robust when you're older. Not saying that your immune system's compromised, but it's just saying it's become more efficient. Everyone already understands or has that college level degree. Now, when they need to do something, when they need to go fight an infection, it's just more efficient instead of housing millions of T cells. I'm not millions, but hundreds of billions of T cells in the thymus. You store a smaller amount of T cells in the thymus, and when they're needed, they go in to the site of infection, and then they divide and get reinforcements at the site of infection, rather than storing a bunch of extra ones in the thymus. Does that make sense, Cliff?
Yeah.
Okay. Well, thank you for the question. But there are lots of other organs, if I can get back to just talking about the immune system. Your tonsils, the lymph nodes, spleen. You also have your appendix. There's also areas in the gut called malts. They also collect immune cells there. Those cells specifically learn to tell what's food, what's not food, so that your immune cells don't start attacking the food in your gut. That's not good for your digestive system, for that to be happening. So there are other areas that we're going to kind of gloss over for the most part.
It's the Roman terror of the immune system: food or not food.
Yeah, exactly. So, I mean, the immune system has to learn all these things, and it takes some time for the immune cells to learn how to do these functions. But luckily, I mean, with the way infants develop, they mostly rely on their mother for their initial immune system. And what I'm saying is the antibodies and things that you have when you're first born, your mother lends them to you through breast milk and just, through the fetal barrier, fetal placental barrier as well. Immune systems like antibodies, some are allowed to pass through that barrier. So the child does have an immune system before any of these cells have the chance to actually grow and learn. But once the baby's delivered, there's a transition, and slowly the mother's protective effects wane as the child develops its own immune system. So any questions about the organs involved with the immune system?
No, it's fine.
Okay.
Sorry, I accidentally jumped to the next one.
No, I mean, this is just the same picture, but a little more in depth. You can see some of the things I was talking about. There are areas called Peyer's patches in the in the large colon that are also, related to making sure that food is identified as food and not as something foreign. You can see there's lymph nodes throughout the body, head, neck, arms, all the way down into your groin. The spleen also removes debris, can remove antigens as well. So all these organs have slightly varied effects and roles in the immune system. And this is just a really nice slide I wanted to include so people can kind of visualize like where the thymus is. This one has a really good demonstration of where it is because most people have never heard of the thymus before.
Yeah, and I would just like to point out, I'm sure a number of people here who have gone to the doctor sometime and had the doctor feel the lymph nodes in your neck.
Yeah.
And as someone, as a matter of fact, one of mine is a little large and sore right now. As someone who has been prone to ear infections throughout my life due to the blasted measles, yeah, that's something I'm very familiar with. But yeah, your lymph nodes get inflamed when you're ill sometimes. I'm sure RSC could explain that a little bit better.
Yeah, we'll get to that. But basically, your lymphocytes are going to travel to the nearest lymph node to quickly repopulate and grow, so they're close to fight where the infection is. And that's why the lymph node becomes inflamed, because they're growing and dividing very rapidly. It starts to stretch the area that little lymph node is in, and as it grows in size, it irritates the nerves that are around the lymph node, causing pain.
In fact, isn't it the lymph nodes that Yersinia pestis ends up collecting in?
Yeah, it takes advantage of that. Mm-hmm.
Yeah, and that's what causes the buboes of bubonic plague, and they can eventually rupture to the outside.
And they can also rupture to the inside, which is also very dangerous as well. But basically, the lymph nodes serve as little barracks, I like to think of, where you have your infantry that is either coming from their headquarters in the bone marrow or the thymus. They basically, their rendezvous point is in the closest lymph node, where they gather up, formulate a plan, and start communicating how to best defend the body. Any more questions from the chat or anywhere else? Otherwise, we can move.
No, don't seem to be any.
Okay.
Everybody seems to be following along quite well.
Or they're busy twiddling their thumbs and just doing something else. But yeah, let's go to the next slide, Cliff. So we've talked about the organs and the tissues that are involved. Now let's take a look at what these cells look like. So a lot of these cells we talked about as definitions earlier. So if we start in the upper left-hand corner, you'll see epithelial cells. On the left, you have cells that protect you from the outside environment, like your skin, like the inside of your gut. Those are just the two classic examples. We also talked about phagocytes, cells that engulf other cells. If you look at them in a microscope, that's almost what they all look like, is very similar. The big blob in the middle with I don't know if you'd call it a star, or it's supposed to represent a virus or a bacteria. It's been engulfed and is being digested by the cell. Dendritic cells. These are one of the antigen-presenting cells. As you can see, they look like they have a lot of roots that extend out. Like Jade said, they have a tree-like appearance. This is what they're referring to. That's why they're named dendritic. But basically, all their extensions are like little arms that feel around to pick up any antigens that are in the environment outside. And they pick up antigens that are self-antigens, but when they encounter antigens that are not self-antigens, those antigens go through the same process. They get engulfed, they get broken down, and then they get displayed on the outside of the cell. On the outside of the cell, they can be used for communication with T cells and other cells that will use that information to direct the immune response.
Are dendritic cells okay against Head and Shoulders? Because I've heard that's good at defeating dandruff. Oh, I forget it.
Dandruff and dendritic, not the same.
No.
Sorry.
No, that's okay. Another type of cell that we're probably not going to talk too much about are plasma protein cells. Those are more related to how you clot and things like that. So they are an important part of the immune system, but we're not going to really be talking too much about them today. And I do want to make a distinction here that when we're talking about the immune system today, we are talking about the immune system outside of the brain. Basically, the names for everything inside the brain are completely different than the names when they're in the rest of the body. So maybe if we have another chance to talk about this, we can talk about how everything is different in the brain versus the rest of the body. But for today, we're just going to talk about the generic body in general. These concepts will all still apply to how the immune system works in the brain, but it's a little more nuanced, and they change the names of everything just to make it more fun for a neurologist and immunologist. So the cell lines we've talked about so far, including the natural killer cells, the ones that will tell virus-infected cells and tumor cells to die or will attack them, those make up what are called your innate immunity. These cells are always present, they are always functioning, they are always performing their task. That's why they're called innate. They act very quickly. You can see on the bottom, there's a scale that represents how quickly these cells will react. I mean, they will, in hours or minutes, they can go in and start fighting an infection locally because they're already at that area. They're everywhere in the body, ubiquitous. So we talked about adaptive immunity earlier. This is where your B and T cells come in. Adaptive immunity is basically what it sounds like. It's an immune response that adapts to whatever the offender is. If the offender is a virus, if the offender is a parasite, if it's a bacteria, your adaptive immunity makes specific attacks against that one single invader, whereas the innate immunity attacks anything and everything that is foreign. So you have your B cells on the top. On the left, it represents the immature or naive B cell. The B cell knows how to make antibodies, but it has to be taught what to make antibodies against. Antibodies are just little particles that can identify an antigen and cling and stick to the antigen. Once they stick to their corresponding antigen, they signal for a phagocyte to come and gobble up that antigen. That antigen could be a part of a bacteria, it could be a protein on a virus, or it could even be a flagella on a parasite. It's all dependent on what the B cell learns to make the antibody against. So below that, you see the T cells. And the T cells are very unique because they are the translator cells for the rest of the immune system. T cells will go pay a visit to a dendritic cell, see if they can identify anything that's foreign. And once they get that foreign signal, they'll go back to lymph nodes, multiply, start excreting their signals like cytokines and interleukins, and tell other cells what to do, what to attack. And you can see the little arms on the T cells. Those are representations of that MHC2 system that I was referring to earlier. That's basically just the arm system they use to figure out what is self and what is not self on other cells that present antigens. So in harmony, the T cells will come in and identify what's foreign, will go back to their local lymph node, amplify their signal by dividing and growing rapidly, expressing their signals like interleukins and cytokines that tell B cells, hey, come here and make antibodies against this antigen. Hey, dendritic cells, hey, phagocytes, come to this area because there's an infection that needs to be fought. And because they're doing a much more complicated and much more precise targeting of a specific invader, this takes longer to do. It takes days for these cells to multiply and have their signal grow strong enough to attract the B cells, the other cells, to come in to the area and do their job and their role in immunity. So this will take a long time. This is why you don't get antibodies to an infection 24 hours after you start feeling ill. This takes days, maybe even weeks in some cases for it to happen. We have any questions so far from live chat or the panel?
No, it's fun here.
Okay. Let's move on to the next slide. So let's talk about innate and adaptive immunity in more depth. We've talked about what cells are involved, kind of how they do everything. But we'll see in some videos later a much more adequate explanation. You do need to see this whole play act out, not in real time, but in in a sort of, animated time to really have a good understanding. But basically, this is a compare and contrast section between innate immunity and adaptive immunity. We see that there's different types of cells that belong to each category. And we've talked about how their activity is, well, the innates, they're everywhere. They're always ready, on alert. Adaptive immunity, they are normally silent. They don't do anything until there is an invasion, until there is some sort of insult against the body. And because the innate immune system, they're always present, always on alert, they're much quicker to respond.
Where because they're, do they say? I mean, do they float around in the blood, or
Yeah, some of them will float around in the blood, but I mean, most of the ones that are going to be fighting something are going to be in the tissues. It's not easy to get infections into the blood directly.
Let me just clarify the question. You said that normally they sit around doing nothing. Where do they sit?
So the adaptive immunity cells, when they're not doing anything, B cells will sit in the lymph nodes doing nothing, and the T cells will normally sit in the thymus.
Okay.
Mm-hmm. Thank you. No, thank you for making sure I cover everything. If I'm not clear on anything, please jump in, because this is a difficult thing and it's a lot to digest all at once. So the innate immune system, like I said, immediate, quick response, but it's very limited in terms of the degree to which it can attack. It's basically a generic response that's not specific to anything. Whereas the adaptive response is a very specific response that specifically responds and is highly effective against invaders. The problem is it takes a long time to build that response up. But here's where vaccination comes into effect. Vaccines trigger your adaptive immune response. They trigger the T cells and the B cells to make antibodies against a certain pathogen. So take the MMR, the measles, mumps, rubella. When you get that vaccine series, your body builds up enough B cells and T cells to have those antibodies on reserve, ready to go should you ever encounter that infection again. So instead of taking the 2-3 weeks it would normally take to build those antibodies, they're ready to go much quicker should you ever become infected with that virus again, and therefore you won't get the infection. You won't become as ill. And most likely, if your immune system is working fully, they will fight the infection before it can take effect. So go back to comparing the two immune systems again, talking about specificity. Adaptive immune system is very specific. This is why they will only target one infection, one virus, one strain of flu. It's very specific to specific antigens of that pathogen, that invader. The innate immune system is like a machine gun. It's just going to spray bullets everywhere and you're going to hit everything, but not hit everything very effectively. So innate immunity, we've talked about that it's everywhere. These cells stay and live in the tissues as part of their life cycle. But they will, if they can't destroy a pathogen for some reason, they'll basically wall off that pathogen from the rest of the body and create a little, almost like a pus-filled sac, basically. We call an abscess once it becomes very large in medicine. But that's how they react. If they can't destroy it, they wall it off from the rest of the body. The adaptive immune response, however, the antibodies are almost always effective against their target. If the antibodies, for some reason, aren't effective against their target, well, I mean, that's the limit of that side of the immune system. In terms of the memory, this is what I talked about with the vaccines. The innate immune system doesn't remember any of the battles it's fought in the past. It fights every battle as if it's the first time they've ever seen this invader, this virus, this bacteria. Whereas the adaptive immune side, the cells that go into making the specific antibodies, the T cells that go on to recognize and coordinate the immune response, they remember that exposure. And if the body is exposed again in the future, their response is amplified much quicker, which is why vaccines are so effective. They basically train your body rapidly to any sort of reinfection by a similar bug.
Yeah.
So how are we doing so far? Everyone's still awake?
Yeah.
Yeah, there was one question in the live chat somebody asked a little bit ago. Is there one kind of cell that is most common with an autoimmune disease?
That's most common, I think, is a bad way to ask the question. But basically, different forms of immune disease are based on the type of cell that's affected. So you can have autoimmune diseases where your B cells can't make any antibodies that work or function. Well, that's going to cause certain defects. You're not going to have a strong adaptive immune system. Your innate immune system will still be fully intact, but the adaptive side will not be robust.
Yeah.
And you're not likely going to take vaccines well in that case.
Yeah. No, he's asking about autoimmune disorders.
Oh, I'm sorry, autoimmune.
Yes.
There are two types of disorders. There are disorders of the immune system where the immune system doesn't function. Autoimmune disorders are basically the cells from your adaptive immunity, or sometimes very rarely in your innate immunity, have failed to distinguish what is self versus not self. And they start attacking the self as if it's not self. This can be either through T cells, through B cells. There's very specific manners through which it can happen. But in most instances, it's through an antibody mismatch. Basically, you have some B cells that have learned to make an antibody against one of the parts that belongs to your body. And this could be because some pathogens will intentionally have signals on their cell surfaces that mimic your body in order to try to prevent your immune system from tagging them and destroying them and fighting them. Sometimes this, unfortunately, causes your immune system to create antibodies that will attack your own body. You can also see this in like Rh incompatibility, which is when the mother gets pregnant with their first child. If they don't have the Rh antigen in their blood, their body has learned to recognize the Rh antigen as a foreign antigen. And if they happen to have a first child that's Rh positive, well, during the delivery or maybe at some point during that pregnancy, their immune system will learn and pick up that Rh antigen and recognize that, hey, this is foreign. They'll start to attack it. And the next baby, if you have another Rh baby, those antigens and antibodies will cross through the placental fetal barrier. And unfortunately, that can cause the baby to be attacked by the mother's immune system.
Yeah,
that's not necessarily an autoimmune disease, but that's kind of the typical features I'm talking about here.
Yeah, that's the category I fell into. I'm rhesus negative. My son was rhesus positive, and with quite a bad 5-hour bleed before delivery, the chances of me being exposed to his rhesus positive cells meant that I would automatically be given what's called anti-D, which mops up these rhesus positive cells to stop me mounting an immune response to rhesus positive cells, so that subsequent pregnancies, if the baby was rhesus positive, I wouldn't already have been primed against them. Problem was they didn't remember the anti-D until day 5 after delivery.
That's late. It's supposed to be given within 48 hours to be.
Yes, I had to ask for it.
There was another question as well, which I've stuck in the bottom of the side chat.
All right, excellent question. So let me read it real quick. Monocytes move from the systemic circulatory system into connective tissues, where they differentiate into what phagocyte cell type? So monocyte is just the definition for what are they called? Neutrophils, which is a specific type of phagocyte. When they're in the blood, they're called monocytes. When they go into the tissues, they're called neutrophils. That's just a naming system, so you know where it is and where it's coming from. I hope that answers the question, and I'm not sure who actually asked it.
Benaberry.
Benaberry. So, Ben, I hope that was a decent answer to your question.
Thank you. Appreciate it.
But an excellent question. That's not very well understood by most people, why we have different names for everything. But basically, it's to track their stage in the process of fighting an infection. A monocyte is a white blood cell, a phagocyte, that is basically traveling in the bloodstream and it hasn't found a home in a tissue yet. So should there ever be an infection in the blood, they will do their fighting there. But their ideal home is to go into a tissue and establish that innate immunity in a local tissue. And that's why they're given a different name, because while they're in the blood, they serve a slightly different purpose. Can we move on to the next slide? What do you guys think?
Yep.
Okay, so this is kind of the story we've been going through today. We've talked about what the immune system is, what it's consisted of. We've talked about a little bit about how it works and how it fights diseases through innate and adaptive immunity. Let's get on to things like how does it get turned on, how does it get suppressed, and how do diseases get around the immune system. All right, let's go to the next slide. So it's very difficult to explain, but like I said, we're going to see a video about this and go through these steps one at a time. But basically, this story is the story that's shared by all types of infection. Some microbe, virus, parasite gets into your body somehow. It gets through that epithelial barrier, your first line of defense. And it encounters what's called the antigen-presenting cells. So these cells will take up the antigens that come from the microbe and digest them and start to display them on their cell wall. You can kind of see that here in the picture. I don't know if you can point to any of these aspects under the number 1 section for me, Cliff Jumper. But you see the dendritic cell. The little star inside is the antigen it's swallowed. And then you see the little, I don't know if you would call them like tree-like or flower-like structures. That's the antigen-presenting portion. And basically, this dendritic cell has encountered an antigen. It can't tell whether it's a foreign antigen or a self-antigen, but it's gone through the normal process. It's digested the antigen and displayed it on its cell wall, cell membrane. That way, other cells can come in and tell it whether these are foreign or not. So that's the first step. The antigen-presenting cells encounter the microbe and the microbe antigens. Now, number 2, we get the lymphocyte activation. So T cells that are just in the neighborhood will come by and pay a visit to this antigen-presenting cell. And they will encounter the little flower, basically, that has the antigen. And through their own little flower or little arm, that's how they read and communicate. The lymphocyte will say, hey, this antigen is actually not a self-antigen. This is where we get the ideas of like opponization and destruction by phagocytes and the activation or the beginning of the communication that the T cell will go. So basically, at this point, the T cell is going to say, hey, this antigen is not something that's friendly, that's not part of the self. It needs to be destroyed and gotten rid of. So T cells will go back to a local area like a lymph node. They'll grow, divide, and start to make their signal happen to start communicating to the rest of the immune system. We'll go down to number 3 now, and this is where the adaptive immune response begins to kick in. Antibody.
What's the big pink ring?
That's supposed to resemble or represent a lymph node.
Right.
So the T cell is in the lymph node. It's multiplied. It's you can see all the little red images that are popping out. Those are supposed to be, I think, cytokines. It's signaling for other cells to come in and start fighting the pathogen here. It's telling where to go. But basically, the B cells will come in. They will start to make antibodies specific to those antigens that the T cell communicates. And this is where you get those large lymph nodes, because you'll have lots of immune cells growing and dividing, communicating inside the lymph node, directing the traffic for all the rest of the immune system. This also I think it says a little bit about how this might be part of autoimmune diseases, where you have T cells or B cells that have learned inappropriate signals to attack, signals that are part of yourself.
Dragnor asked, what is a lymph? Explain what a lymph is and the network around the body.
Well, basically, you have the blood network. I think most people are familiar with arteries. Arteries carry oxygen-rich blood from the heart, and you have veins that carry blood that has less oxygen back to the lungs and the heart. Lymph is the third fluid system in the body. It carries the excess tissue, moisture, liquid, and it also is the transit system for the immune cells, because not all the immune cells go in the blood. Cells like your lymphocytes, they basically stick mainly to the lymph system, and it allows them to travel throughout the body as well. And the lymph system generally follows along with the blood vessels. So you'll see the lymph system next to arteries and veins throughout the body. It's basically the drainage system, the sewer system for your body, for lack of a better word.
Sounds a little bit like the internet or Twitter.
A little bit. That's kind. That's kind.
All
right. Any other questions? I thought that was a good question, Drag. All right, keep going. Let me know if there any more questions pop up, and we'll jump to the next slide. So, in the explanation again, just in these stages, first antigen of the disease is detected. Second, you get that clonal expansion, which is just a fancy way for saying the cells that have detected the disease start to grow and multiply and start signaling for backup. Then you have what's called differentiation, where your B cells will come in. They will differentiate into populations that will make antibodies specific to one antigen that target one aspect of the pathogen, that target one aspect of the invader. And then the antibodies that B cells produce go out, attach to their appropriate antigen, and tell the rest of the cells, hey, this is where you need to attack. The phagocytes recognize these antibodies and start to chew away and chow down, basically, until the infection is eradicated. Or if they're unable to eradicate it, they wall off the infection from the rest of the body. And this basically will prevent disease and death in most cases. And then in the final stage, you have the memory. The T cells and the B cells that were activated as part of the adaptive immune system, they retain that function for the rest of their lives, and they pass it down to their daughter cells. Once a B cell has differentiated or matured, it only makes that one antibody for the rest of its, the rest of its life, and all of its descendant cells will only make the antibody for that one specific antigen indefinitely, and that lineage will survive in your body in a lymph node, or maybe even in a tissue somewhere. And will provide that memory, that more rapid response should you ever encounter the same disease or infection again in the future.
Donica has a question saying, with some exceptions, vaccines seem to be better for viruses that for bacteria, e.g. tetanus vaccine, is aimed at the toxin, not the pathogen. Anything to say about it?
Yeah, this probably would have been a good question for Ian if he was here, but yeah, the tetanus vaccine, the problem with tetanus is specifically the toxin, not the fact that your body can't get rid of the bacteria. The problem is that if the bacteria is able to establish a foothold, the toxin prevents the body from effectively fighting the infection. So if you effectively eliminate the toxin by making antibodies against the toxin, the body can easily get rid of the pathogen, the bacteria in the first place. That's why that's more effective for bacteria. The other thing is the bacteria, they're generally larger than viruses. They generally can only get into certain areas, direct contact. So the route for exposure is a much more nuanced for bacteria. There's only certain ways you can acquire bacterial infections, usually through like abrasions on the skin, like a cut, or directly through an abrasion on like an inside of your mouth or something. It's very rare that you would get anything that comes through a different route. But since bacteria are larger, it's much easier for the innate immune system to identify them and pick them off immediately. It's the viruses that are a little more tricky because they're so much smaller. They have an easier time getting around those large phagocytes. So that's why vaccines are mainly directed at, that's probably not the main reason, but mainly we need the vaccines to help us prevent viral infections, because they are much more easier spread, and they are much more easily passed by, they much easier pass by the innate immune system and rely on that adaptive immune system as the primary mechanism to fight that infection. And we know that it takes weeks in a lot of cases for that antibody response to develop. So you're giving the virus 2 weeks of a head start in terms of getting a foothold in an infection when you're going through a normal immune response. So we'd rather get around that and make that immune response much quicker to prevent disability, morbidity, and death. That's why most vaccines target viruses instead. Bacteria, yeah, in some cases, like the tetanus toxin, we do want to target the bacteria. Whooping cough, we want to target the bacteria that causes whooping cough, because we want to prevent those infections. But the innate immune system usually is very good against the bacteria, just as an overall general expression.
Yep.
So it has to do by the timing. We want to try to make the timing as short as possible, give the body the ability to fight the serious infections much quicker. And unfortunately, most of the serious infections are viruses. It was a great question. Anything else that you guys have seen in the live chat?
No, it's fine.
Okay. I think we're getting close to the end, and we're getting ready for some videos. So let's go to the next slide. Okay, so this is a nice graph, kind of giving you a timeline of how things go. The antigen recognition phase, I mean, it takes a couple of days for this phase to really go through until you get the activation of the lymphocyte. And this is why I'm saying that it's more important to get vaccinated, because you reduce this time. Lymphocyte activation happens next, and you can finally get activation, and you can start getting rid of the infection in the antigen elimination phase. But you see that you have to wait more than a week for any type of that response to occur. And eventually you get to the stage where the infection is cleared on the very right, which is weeks later, and your body retains the memory of that exposure should you ever be exposed again to the same infection. Are we still good? Everyone's still following along?
I don't know if Fiona wants to address something that Tommy brought up.
No, I think we should just keep going. What do you think, Fiona?
If she's talking, she's...
Sorry, I was pasting something there, and it's just disappeared completely. Oh, yeah, no, it's there. It's there. I see it now. Yeah, Tommy said the innate immune system does actually have memory.
I'm not sure what he means by that, so I don't even know if we can address it, because if his definition of immunity and memory are something completely different from what I'm talking about, I can't really address whatever he's talking about.
Well, you
want to jump into the weeds here, or want to keep going?
Well, if I remember correctly, you were saying there's the innate system and the, what was the other one called?
Adaptive.
Adaptive, yeah. So effectively, the adaptive one, it gains the memory, and the innate one doesn't have the memory because it starts off clean and then learns something. Learn something through the adaptive one. If I'm correct, that was what was being explained.
Well, Tommy,
yes.
Tommy has clarified slightly and said, "Google it."
Yes, you see, Googling is a great way to do things. Google it can lead you to like weird places like Natural News, which is just silly sometimes.
I'm just trying to find out what Tommy's actually meaning in the use of his terms.
I don't think he knows.
I don't think Googling will give us an answer to what does Tommy Hall mean when he says innate immune system has memory. I don't think that's going to be a search that will grant anything.
Yeah.
My understanding is when we're talking about B cells having a memory. That's just an analogy.
Yeah.
What we're
Yes.
talking about is them obtaining a chemical shape.
You're roboting again, Jude.
Crap. Let me type it.
Okay, well, Tommy has posted a little phrase here. Immunological memory is no longer a unique trait of adaptive immune cells.
What?
That's great, but I still don't know what you mean by that. Unfortunately, I'm not getting a description of what you are calling immunological memory, Tommy. So if you want to give that to us, I guess we could talk about it. But I'll give you a couple minutes if you're able to produce something.
Okay. Can you hear me now?
Yes.
Yes.
Okay, I'm okay. So what I was trying to say is when we're talking about, as I understand it, when we're talking about obtaining a memory is just an analogy.
Mhm.
What's actually being talked about is obtaining a chemical shape. And the example that was used back when I was in elementary school was the whole lock and key example.
That's actually a very good analogy. The antibodies are the keys, and the antigens are the locks. The B cells, it's not necessarily that they have a memory of the pathogen. They learn only one key to make, and they make that key forever in their lineage.
Yes.
That's
Right,
the
and they-
memory we're talking about.
Yeah, and they make that key because they've run into something else. It's a bit like they're a bit like plastic. I don't know if this is a good analogy or not, but kind of like Play-Doh, that it's not hard, except it's not, or clay that's not hardened yet, and they run into something, they obtain a shape, and then they maintain that shape, and because they have that shape, they're able to reproduce that shape.
Yes, I think it's a nice analogy. But like I said, I think maybe we'll all benefit once we get to see this stuff in action.
Yeah,
It
yeah.
really only makes sense when you see it. It's very hard to describe.
Fortunately,
So,
we're having to use analogies, which is not always a good thing for some people.
Yeah, and I understand that Tommy's saying that this is a title. Unfortunately, the title doesn't tell me what you mean when you say immunologic memory. So if you have a definition of immunologic memory, feel free to post it, and we can perhaps discuss it. But I think we should just move on for now.
Yeah.
So let's go to the next slide. I think we're wrapping up. I didn't put it together a lot. So here for I think was it Benna that asked about autoimmunity, or who was that asked the question about autoimmunity?
I think it was Benna, yeah.
Okay, well this is more in depth. There's basically four major types of what we call hypersensitivity or autoimmune reactions. And this is where I said it really depends on what's causing the dysfunction. They basically just labeled them 1 through 4 based on their discovery. So the first one discovered is the type 1. This results from a very specific type of antibody called an IgE antibody. These come from a very specific type of B cell called a mast cell. So basically there are subtypes of B cells, which we didn't even talk about because, like I said, I wanted to keep this very superficial, very basic. But they make this specific antibody called IgE, very effective for some instances, but unfortunately gives you that allergy reaction where you get the runny nose, the redness. That's what this antibody causes. And basically in a type 1 hypersensitivity, you have an allergic response to some particle like pollen, let's say, as an example. And these cells overreact into the presence of pollen. And they cause you to have all those classic symptoms, the watery eyes, the runny nose, the cough, the congestion, things like that. We call this seasonal allergies for the most part. That's the type 1 reaction. It's those specific type of cells called mast cells. So the second type is through an antibody response. The second hypersensitivity is basically when you have antigens that occur against something that's on the surface of a cell in your body. It can be any type of cell, but basically the antibody is going to attach to that cell and cause inflammation until the phagocytes, hey, attack here. So it unfortunately leads to phagocytes attacking your own cells, causing inflammation, and in some cases, pain, discomfort, and dysfunction of whatever's being attacked. So it's the general hypersensitivity type reaction can cause a wide array of symptoms depending on what's being attacked. If it's heart tissue that's being attacked, you're going to have heart problems. If it's muscle being attacked, you'll have muscle weakness. If it's bone, you'll have brittle bones. You can get fractures. It can literally happen against almost any cell type. And depending on what cell's being attacked, that tells you, or that will determine what symptoms you show.
Can I just ask a question for a second?
Of course.
So in the UK at the moment, we've got a guy that was hit by, I don't know, he's been infected somehow by oh, cracky, what's it called? So like sarin is an example of it.
Oh, you're talking about the former Russian spy who was,
Yeah.
his daughter were attacked by a nerve agent.
A nerve agent, thank you. That was what I was thinking of. And one of the, I remember there was a doctor talking about it today and saying that one of the things these nerve agents can do can attack the muscles in the heart and things like that, which causes them to shut down and stop functioning properly. Is this where the immune system is compromised and unable to fight such an infection?
With an autoimmune condition like these things that we're talking about, type 1, type 2, type 3 hypersensitivity, it's completely unrelated to the body's ability to fight an infection. So I talked about earlier where there's two types of immune dysfunction.
Mm-hmm.
One where the immune system doesn't work, and the second is where the immune system attacks your body. So these four types of hypersensitivity are a specific subgroup of autoimmune dysfunction, where the immune system attacks your own cells or acts abnormally and causes dysfunction with your own body organs, tissues, etc.
Yeah.
Does that make sense?
Yeah. I'm just wondering, with the gravity of this nerve agent thing, is the reason why the guy's suffering so much, and his daughter, and also a police officer as well, who got cross-contaminated, is the reason that they're suffering because their immune system is unable, hasn't learned what needs to be targeted, hasn't learned hasn't learned a safe to the key to break into the affliction, I suppose you could call it.
Yeah, I mean, it really depends on what they were exposed to, but they've already denied that it was sarin gas. So I could tell you about sarin gas, but that's not what it is. It depends on what the actual agent is. But agents of like chemical weapons, they don't generally behave like a pathogen. Chemical weapons are very effective because they will get absorbed readily into the body through, mouth, skin, whatever. And then they go right to work immediately. There really isn't any downtime for most nerve agents. They act very quickly. They get absorbed into the cells rapidly, and the immune system really has no way of defending against these type of chemical weapons. You have to really rely on your body's detoxifying system, which involves the liver detoxifying the chemical, making it more water soluble, and then you piss it out. That's the way your body fights this kind of chemical exposure. It's not at all related to the immune system, unfortunately.
Okay, right.
Unless it's some sort of biological chemical, which, I mean, that's way above what we're talking about today, Cliff.
Yeah.
But a very interesting question. I mean, we can speculate. I don't know if they'll ever tell us what it was that they were actually exposed to, though. State secrets and all.
Yeah, but again, it'd be getting off the topic.
Mm-hmm. So just a quick recap. So we talked about the first type of hypersensitivity, seasonal allergies, comes from specific cells, mast cells. Things like Benadryl work really well at preventing those cells from overreacting and dumping out their IgE and causing you to have all those weird symptoms. Type 2 hypersensitivity, different. This comes from antibodies that are created by some abnormal B cells that have targeted something in your body. It's part of you. Causes dysfunction, local dysfunction, wherever they're making those antibodies. So type 3, this is where people get very confused. So I hopefully will explain this very well in the first go around. This is also antibody related, but it's not just antibodies. It's antibodies that clump together into complexes. So basically, we have some antibodies that are made by B cells that are extra sticky. They will stick to each other. It's the fact that they stick to each other and form a big glob of antibodies. This collection of antibodies will go and cause immune problems, inflammation, pain, and tissue destruction, more specifically to your blood vessels or your joints. So you get what's called vasculitis, which is inflammation of your blood vessels, or you get arthritis, inflammation of your joints. And this is because the antibodies clump together and basically disrupt the normal flow of blood or disrupt the normal function, mobility of a joint. And then your body starts to respond by causing inflammation and having an immune response locally to try to clear out those complexes. But again, you have phagocytes that can only do their random process of innately destroying what they see and what they feel, and it's very non-specific. And your adaptive immunity has no role here because they have caused the problem in the first place. So how did that explanation sound? Does everyone kind of get that? The difference between type 2 and type 3? They're both antibody related, but type 2 is specifically just the antibodies directly causing the effect. Type 3 is basically the antibodies sticking together, and the fact they stick together causes the defect.
Yeah.
Okay. Good. Type 4. This is a slower type of hypersensitivity. It's called delayed hypersensitivity. But if you've ever been exposed to poison ivy, this is the classic example of type 4 hypersensitivity. Basically, you have an inappropriate immune reaction that's caused by your adaptive immune cells, specifically the T cells. The T cells recognize something that they probably shouldn't think is harmful, and they tell your body, no, this is very harmful. Attack here, attack now. Also, you can see things like Crohn's disease, and they think even MS, multiple sclerosis, is a type 4 hypersensitivity. I don't know if they've proven that yet, though, so that's kind of speculation in my mind. But it's delayed. This doesn't happen very quickly because we understand that T cells, they take a while to build up that immune response. It happens after days or even a week. They're able to mount the immune response. And something like poison ivy, you rub up against the poison ivy on a part of your skin, the T cells in that area eventually will get a hold of that antigen from the poison ivy and say, hey, start attacking here, start making antibodies to this. It's all directed through the T cell, and it happens days, weeks later, which is why a lot of times when people get exposed to poison ivy, they don't even realize they've been exposed, and it's not until a week or two later, after coming back from their camping trip or what have you, they start to break out in that rash and start itching and start having the pain. So, type 4 hypersensitivity. So these are the autoimmune diseases and their patterns of effect. Any questions so far? Or are we still good?
Well, other than Tommy saying that fasting works wonders on MS, which...
Yeah. Okay. Classic. Tommy, your stick never gets old, I guess. In some corners of the internet. Let's go on to the next slide then. All right. Now we get to actually watch some of this stuff happen in real time. Can we start with the first video?
Yep.
Not that first video, but the first YouTube video.
Yep. Okay. Remember that I have to turn my audio up a bit. So if anybody speaks, you're going to be really loud.
Yeah, just make sure you pause like after 30 seconds or something.
Yep.
Our immune system is made up of two distinct types of immunity. There is the innate immune system, and then there is the acquired, or also known as the adaptive immune system. The innate immune system is concerned with keeping harmful invaders from penetrating into the body. The adaptive immune system is a more complex response to invaders that do penetrate into the body, primarily through an inflammation response. Let's begin by looking at innate immunity, which is something we are born with and does not continuously change, as does the adaptive immune system. Innate immune protection is always present in exposed parts of the body most vulnerable to the outside environment, such as our eyes, skin, nose, mouth, lungs, and the digestive system. Mast cells and natural killer cells play dominant roles in our innate immune defenses.
All right, stop.
Yep.
Okay,
Stop.
very good. Everyone following along? A very nice artistic depiction of the cells and the types of invaders they encounter. And like they said in the video, the innate immune system is the 24/7, the 7-Eleven of the immune system. Doesn't take any holidays. It's always working, always present.
Continue to play.
Yeah. If no one has anything they want to add, let's keep going, I guess.
Type of immune
I think you can finish up the video.
is our acquired.
Yeah. The next one.
I think you can finish out this video now.
Oh.
That's all right.
I'll have to go and find it. Give me a sec.
Nice explanation of the two systems, and they talked about the innate immune system, and now they talked about the adaptive or acquired immune system.
Okay.
Our innate immune defenses. The second type of immune protection in the body is our acquired immunity, which is also known as the adaptive immune system because it has the ability to adapt and recognize and fight new antigens. Because our environment is made of an endless number of invading antigens, which are constantly evolving and changing, it is essential to our survival that we have a defense system that is capable of adapting to the changing threats as they occur. When we encounter a foreign substance, our adaptive immune cells jump into action. Unique white cells are then developed to identify and destroy the invading substance, as well as remember it for the future. This process in the adaptive immune system is called immunization.
It's worth pointing out that
Unique.
Don't eat worms, okay?
Don't eat worms.
Yeah, don't eat worms. That looks like a worm to me. Anyway.
It looks like a tapeworm. Unfortunately, I don't think most people choose to eat them. It's accidental. But it still happens. It's still very much a problem. But your body does have an immune system that can defend against those kind of pathogens to a degree. Unfortunately, most of the time you need antibiotics or anti-parasite drugs to get rid of them. So anyone have questions on that first video? The second video, I think, kind of rehashes it in a little more detail. So let's pull up that second video if we can.
Yeah.
An immune response involves events that unfold both locally at the site of an infection and at more distant sites, such as nearby lymph nodes. We can see the integration of the different parts of the immune response if we follow the course of a typical infection. Most pathogens are kept outside of the body by epithelial barriers, such as the epidermis, and are crossed only when there is an injury or tissue damage. After an injury, bacteria cross the epidermis and establish an infection in the underlying tissue. Phagocytic cells in the tissues, such as macrophages and neutrophils, engulf the pathogen.
Okay, pause for a second. All right, so this is just a point of clarification. The macrophages and the neutrophils are just two types of phagocytes. This video is offering a lot of good details, but don't want to try to overwhelm people. These are the phagocytes I was talking about. There are many different types. We're not going to get into all the types today.
What were the little red things again?
That's supposed to identify the infection. So the big red ones that are in the skin or in the tissue are the infection, and the little ones that are on the outside of the macrophage, that's antigens that the macrophage is presenting so that the T cell can come and read it in the future.
Right. Press play.
Go ahead.
Dendritic cells are also phagocytic and are activated by binding pathogens to leave the site of infection and migrate to a lymph node. The migrating dendritic cells enter the lymphatic vessels and are collected in a draining lymph node. In the lymph node, T cells are activated by antigen presented by the dendritic cells, and in turn activate B cells to secrete antibody.
Okay, pause it.
Yep.
All right, so we saw the blue circular cell. That's supposed to represent the T cell. Yep. And then the other circular yellow cell is the B cell. And you see the little Y-shaped antibodies that it produces.
Yep.
Yeah, go ahead.
Effector T cells and antibody molecules return to the circulation. They leave the circulation again at the site of infection, where inflammatory mediators have induced changes in the blood vessel endothelium. CD4 T cells activate macrophages to become more cytotoxic, while antibody recruits complement to lyse bacteria directly and to opsonize them, enhancing their uptake by phagocytes. In the case of a viral infection, activated CD8 T cells would kill any infected cells present.
Okay, I hope everyone enjoyed that video. That really nicely translate all the things I've been trying to explain.
Yeah.
I don't think that it's very easy to do with without a lot of images. Questions? Anything else from the chat or the panel?
No, that's making a lot of sense.
So, Cliff, you think those videos are helpful?
Yeah, I do.
Okay, good. If you're on board, let's go to the next video.
Okay.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction.
Can you pause it?
Yep.
Okay, thanks, Cliff. So I hope everyone can see the little arms. This is where we get that idea that there's a lock and key mechanism. The blue T cell here almost has a little like key shape in its extension there that only fits one type of antigen. And for this infected cell that's on the upper right, it just happens to have the right key that fits that antigen that it's displaying. So the infected cell is showing the right type of antigen or has the right type of lock, and the T cell has the right type of key. And when those two come in contact, the key of the T cell unlocks the toxins and the signals that kill the infected cell.
A question for you. Does it kills the cell here? Could it be dangerous if effectively there's too many infected cells and too many cells being killed?
Yes, that's actually quite possible. I think a good example of that would be what's called fulminant hepatitis. Sometimes when people get infected with the hepatitis virus, the infection becomes widespread and the immune response becomes very robust, attacking all the cells that are infected, and it effectively shuts the liver down. That's very deadly because without a functioning liver, you'll die in 2 to 3 days. So that's a critical life-threatening condition where the body's response to an infection is the real problem. It's the life-threatening condition. Because the hepatitis people live with hepatitis all the time.
Yeah, we brought this up in a previous hangout. I think it was in one of the flu hangouts when we talked about a cytokine storm.
Yes.
Where the immune system just overwhelmingly responds in such a way that it actually causes you an awful lot more damage. And in fact, in quite a few cases of meningitis, it's part of the immune response that's responsible for the disseminated intravascular coagulation, which has the end result of having to have limbs amputated.
Yeah, and then we're seeing the same type of effect in the influenza outbreak this year. People with healthy immune systems who didn't get vaccinated are more likely to have this overreaction where the immune cells in the body are identifying and causing a cytokine storm or an over amplification of the immune process to get rid of the influenza virus. They have the good intentions to get rid of the infection, but unfortunately they're doing it at such a rapid pace that it causes the body to have dysfunction. And when the body has dysfunction, that's when the chances of death and disability are elevated. And that's what we're seeing this year with the influenza strain, unfortunately. And it's all due to the immunology behind it. And unfortunately, Ian's not here to discuss that. But we at least have given a nice overview. So hopefully we can have him in a future hangout go more in depth on it. Do we have anything else? Fiona, that was an excellent comment. Thank you.
I do my best.
You do an excellent job. And Cliff is our projector director. So if we want, I think we could finish this video up.
Yeah.
Of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Interesting.
Fiona, any thoughts on that video?
I'm sorry, my mind was elsewhere.
That's okay.
Trying to deal with a live chat.
You deal with them. Do we have any questions that I can respond to? If not, we can just keep moving. I thought that was very succinct, and I think everyone could kind of relate and follow along with these very nice diagrams and explanations, which is why I chose these videos.
Yeah.
All right, Cliff, you're doing a great job. Let's go into the next section, which I think is about how the body's immune system works on cancer.
Our immune system uses antigen recognition to detect both external threats, such as pathogens, and internal threats, such as cancer cells. When healthy cells transform into cancer cells, they may begin to display abnormal tumor-associated antigens. The immune system can detect these antigens and tries to mount an immune response to eliminate cancer cells. But sometimes cancer cells evade recognition and elimination by the immune system. The relationship between the immune system and cancer is characterized by 3 phases: elimination, equilibrium, and escape.
Pause.
The first phase is elimination.
Oh, can you pause?
Yep.
All right. So we never really got into the phases associated with, carcinogenesis and the immune escape, but I think this is the video here that can maybe answer Tommy's question that he had earlier about how does the immune system get tricked. Well, like we were saying, trick is an analogy. Basically, mutations occur to the point where antigens that are displayed by the cancer cell no longer cause the immune system to react. And that's when they evade the immune system. And we'll see that again here in these 3 steps. But was there anything I could clarify so far? Jade or Cliff or anyone in the chat?
No, it's fine.
Okay. Let's keep going.
The first phase is elimination. This is when the immune system detects the presence of cancer cells and tries to remove them. Activated T cells play a major role in eliminating cancer cells through the release of enzymes and other chemical messengers. Mature B cells release antibodies that neutralize or mark the cancer cell for destruction. The second phase is equilibrium. This is when some cancer cells still persist, but the immune system is effective enough to prevent tumor outgrowth. The third phase is escape. This is when some cancer cells escape immune system detection or elimination by interfering with the regulation of the immune response. By evading the immune system, cancer cells are free to grow into clinically apparent disease. In fact, many tumor types have been observed to modulate the immune system to prevent an immune response against cancer cells.
It makes you wonder how often people get cancer and it gets killed off by the immune system before it can really start to get a hold and propagate.
Yeah, I mean, we've talked about this, the examples we've always said, your body always has like maybe 1,000 tumor cells in it at any given time. Yeah, but those cancers are locally controlled, and the immune system's very effective. 99.999% of the time, it's going to eradicate and eliminate the cancer, the tumor, completely. It doesn't get a chance to reach equilibrium or escape, as demonstrated in the video. But because we have so many cells, even 99.999% effectiveness means that 1 in 10,000 cells, 1 in 10,000 cancers is going to go on to that next phase, the equilibrium phase. And even if 99% of the cancer cells that make it to equilibrium are destroyed and eliminated, well, there's still that 1% that can still make it onto the third phase of escape and go on to develop into a tumor or a cancer. And because you have trillions upon trillions of cells in your body, the odds are that if you live long enough, you're going to develop a tumor at some point in your life. And that's just strict mathematics. Unforgiving mathematics. Questions from the chat or anyone else so far?
None so far.
No, lots of comments going on, but nothing of actual relevance to the videos.
So we either have everyone's completely distracted, everyone's following along perfectly, or everyone's clueless. So let's pick that we're doing a great job so far. And let's watch this last video.
Okay, this one's a long video.
Many genetic and environmental factors can cause tumors to form. For instance, UV radiation from the sun can damage DNA and other structures in melanocytes, which are the pigment-producing cells in the skin. Chronic damage to melanocytes by UV radiation leads to most cases of melanoma, which is a type of skin cancer. As melanomas grow, they can eventually spread to other sites in the body, such as the lungs and liver. The cells of the immune system are continuously monitoring our tissues. Natural
pause it for a second.
Yep.
All right. So I just want to make sure that we're breaking this video up a little bit. But we can see the innate immune cells here that are always present, that are always kind of doing at least a baseline level of detection and protection. It's very hard for them to figure out what's foreign versus self. It's much more difficult when one of the self cells becomes a cancer. These cells aren't specific enough. They don't have that specificity to make that distinction. And this is why I thought this was a very good example of why carcinogenesis is so difficult for the immune system to detect. Well, the innate immune system that's always on surveillance, that's always on patrol 24/7, it's very weak at recognizing self versus non-self. It's very good at detecting like bacteria and fighting, engulfing, those kind of things, but it has very weak ability to do the job of fighting cancer. Now, the specific qualifiers, that the natural killer cell that you see here, that is actually one of the more specific cell types that is available to do that cancer recognition and the cancer killing effect. But as we'll see here in the video, that's not 100%. All right, let's keep going.
NK cells sense stress-associated molecules on damaged and cancerous cells. Dendritic cells, or DCs, activate cytotoxic T cells, which can then sense tumor-associated antigens using their T cell receptor and other co-receptors. Once activated, NK cells and cytotoxic T cells release perforin and granzymes. These molecules punch holes in the surface of the tumor cells, causing them to die by apoptosis. Helper T cells support these responses. They help DCs to activate cytotoxic T cells, and they produce cytokines such as interferon gamma that recruit and activate more NK cells. As the tumor evolves, genetic changes occur that can give some tumor cells a survival advantage. This means tumors are often heterogeneous. For instance, tumor cells may no longer express the molecules that are sensed.
Okay, so there were a lot of terms and other things that were kind of flown around. I just wanted to clarify. So interferon gamma is just another cytokine, another cell signal. And you could see in this video that these cells are making contact with each other. That's how they communicate, how they network in real life. That type of networking is what allows them to direct where they're going to have their effect. And the statement, the heterogeneousness of the cancer, that just means that the cancer cells are not all completely identically similar. There are different populations within the cancer, genetically speaking. So some cells may have gene big A, some cells may have gene little a. They're not homogeneous, which means identically the same. Were there any other terms that could use clarification so far?
No, it seems fine.
Okay, let's keep going then, Cliff.
By the killer immune cells. As the immune system continues to kill the tumor cells it can recognize, the cells it can't sense become more prevalent. This is immunoediting. It leads to the emergence of a tumor that can't be detected by the immune system. Some tumor cells actively suppress T cells by expressing inhibitory molecules such as PD-L1. PD-L1 binds to the PD-1 receptor on T cells and deactivates them. This is an immune checkpoint. In addition, tumor cells can attract immune cells that suppress the activity of other immune cells, thereby supporting tumor growth. These immunosuppressive cells include regulatory T cells and certain types of myeloid cells.
What are myeloid cells?
Myeloid cells are basically just another form of specific lineage of white blood cells. they'll become what are called macrophages or neutrophils in the end. But myeloid just is like a umbrella term used to refer to all those types of cells and all the descendant cells. But they specifically come from bone, or myeloid, which is another term for bone.
Right. Okay.
Therefore, the tumor microenvironment is like the scene of a battle between two opposing immune responses. One side of the immune system is attacking the tumor, while the other side is helping it to grow. Scientists are developing immunotherapies to help strengthen the immune attack. One approach is adoptive T-cell transfer. Cytotoxic T-cells taken from patients with melanoma are selected for their ability to attack tumor cells. The best killers are infused back into the patient. Treating patients with cytokines, such as interleukin-2 and interferon alpha, can also boost the activity of anti-tumor immune cells. An alternative approach is to target the immune checkpoints. For instance, antibodies that bind to PD-1 stop this molecule from switching off cytotoxic T cells.
A question, why is this one kind of turning into a Pac-Man ghost and sort of sucking onto it as opposed to the other ones, you know, the other ones that just seem to just grab onto the bits coming off it? I can't think what you call them again.
No, this is just a animation and creative licensing. Basically, they've made it a little more. I would say this is probably closer to what you would actually observe were you to sit and stare at cells interacting in vitro. They have to bind through a variety of receptors and regulators. Basically, the receptor is like the key lock mechanism. So the cancer cell will have the lock, the T cell will have the key, but these don't trigger any event. They just literally allow the T cell to adhere to that cell. So you need to get a certain amount of binding between the cells before they can actually perform this function of killing the cell. And there's several mechanisms that are related to that. But the main one they demonstrated was the MHC1 and the PDL1 as the off switch kind of in this cartoon. But it looks more like a Pac-Man. That's just, I think, a creative license on their part. I don't think it would actually look like that in real life. But they definitely do this sort of dance where you see one cell kind of kisses the other cell, and it's like a kiss of death.
Mm.
Another immune checkpoint being targeted is CTLA4. Blocking this molecule helps DCs to drive anti-tumor T-cell responses. Not all patients will respond to these immunotherapies, and some responses will be delayed. Combining immunotherapies with chemotherapy or radiotherapy can lead to a better response in some patients. Immunotherapies can themselves be combined. For example, PD-1 and CTLA4 blockade can improve responses when administered in combination. Activating the immune system has risks. Some patients develop harmful side effects when their immune system attacks healthy cells. Nevertheless, there have been encouraging results from clinical trials. This animation has focused on melanoma, but it's becoming clear that immunotherapies can be used to treat many different types of cancer.
All right. I thought that was probably the better of the two cancer videos, but I thought the first one was a little more of an introduction video. The second one definitely had a lot of medical jargon, a lot of cell biology in it, and hopefully if Fiona was watching, she appreciated that.
I did. I really liked that last video. It's an excellent video, and yet it simplifies it so much so that anybody can grasp it.
Yeah.
And again, the Space Invaders is an analogy. There aren't actually Space Invaders going on in your body.
Yeah.
But it's a great way to kind of break it down to something that we can relate to.
Yeah. I think that's half of the trick. This is why we use analogies so much, because we're trying to explain some really complex processes, and without a frame of reference, it can be difficult to get it across. So you pick an analogy where people will have a frame of reference and they can grasp the overall picture that way. And then you can get into the intricacies of it. But until you have the shape of the overall topic in your head and know where to fit in the detailed information, then the detail just goes whoof over your head, plop onto the wall.
Yeah, you're definitely right. And I mean, it's very nice to be able to break it down into such small segments. Like we're just looking at one or two cells behaving all at once. We have to realize that in the body, it's actually millions of these cells all interacting in one area at the same time in basically a mosh pit of madness where chemicals are being signaled between cells, chemicals are being excreted to the blood to signal other cells to come join in the fight. And all this is happening at the same time at a much more accelerated rate than what we're looking at here. So breaking it down, slowing it down, minimizing the scope is really the only way to digest this in any form. And with enough exposure, with enough, medical jargon and background, eventually you can come to an understanding that is applicable. And that's what we're looking for, is to get people to understand why these concepts are useful in medicine and health. Why we want to give patients chemotherapy, immunotherapy, why we want to give people vaccines against toxins and viruses, is because we've documented these effects. We've studied them very well for almost 10 plus decades in some cases. And now we're using that knowledge to basically hack the body systems, take advantage of the systems we can use, take advantage of the defects in the pathogens, the viruses, the bacteria, or even the cancer, informing cures and therapies. So that really wrapped up my part. I was hoping Doc would be here to give a little section on vaccines, which is why I didn't go into it in depth. But maybe we could just break it up and do a little free talk. Fiona, Jade, if there was something you thought we should cover.
I don't know if you guys want me to do this. Oops, unplugged myself. I have a really terrific section from a book. It's called Predator. What is it? Yeah, Predator Rex. Predator Rex, I think. That talks about what happens when the immune system goes to work. It's actually pretty cool. I don't know if you're interested in me reading it. It's several pages, but
I'm for it.
Yeah, I am as well.
Okay, so this is a fantastic section, and please interrupt if you have any questions or if you have anything to add to the narrative. One thing I would mention is the first thing it does at RSC, you might explain this a bit, is it talks about the complement molecules that show up, the complement system. So I thought you might, there is,
Jade,
hang on, I don't know if,
Jade, you're brushing your mic against your clothes or something or other?
Oh, okay, sorry. No, I was, that's okay. I was moving it forward to try to get this. Hang on. This is a, God damn it. Sorry, when you get a comment, it covers up. Anyway, that's a little Wikipedia thing on the complement system. So anyway, I'll just go ahead, but please, please interrupt me. So am I sounding okay now?
Mm-hmm.
Yeah, that's fine.
Okay, so this is, from a book called Parasite Rex. Inside the Bizarre.
Actually, Jay, just before you start, you've obviously got the external chat open. If you go to the top where the cog is, you can turn off notifications or stop it blinking.
Oh, okay. The external chat. Yeah. Where is the bleeping cog? Wait. Options. Notifications. Yeah, thanks. Good idea. Okay. So anyway, whoops. Accidentally went backwards with it. I'm doing this on my Kindle, although I have the actual book. The book is really excellent. It's called Parasite Rex, Inside the Bizarre World of Nature's Most Dangerous Creatures.
Yes, I've heard of that book as well. I have not had a chance to read it yet, though.
Now, it says page 57, but this is on Kindle, so the numbers are not exactly. So let me start. The immune system attacks an intruder, bacteria crawling into a cut, for instance, in a succession of waves. One of the first waves is a collection of molecules called complement. When complement molecules hit the surface of bacteria, they latch on and change their shape so they can snag other passing complement molecules. Gradually, the molecules build up on the surface. They assemble themselves into tools of destruction, like drills that can open a hole in the bacteria's membranes. They also act like beacons, making the bacteria more visible to immune cells. Complement molecules also land on our own cells, but they do no harm. Our cells are coated with molecules that can clamp onto a complement molecule and cut it apart. Also arriving early at the cut are wandering immune cells, the most important of which are the macrophages. They have some crude ways of recognizing bacteria. If they happen to bump into them, and they can suck the invaders into their cores and slowly digest them. At the same time, the macrophages also release signals that bring the rest of the immune system's attention to the site. Some of these signals make the infection swell up by loosening the neighboring blood vessel walls. That lets other immune cells and molecules flood into the tissue. The signaling molecules released by the macrophages also latch onto immune cells that happen to be flowing by in nearby blood vessels. They lead the cells through the vessel wall and to the infection, like a boy dragging his mother by the hand down a toy store aisle. With enough time, the immune system can organize a new wave of attack using much more sophisticated cells, B and T cells. Most of our cells come with a standard issue of receptors on their surface. One red blood cell looks pretty much like the next. But when B and T cells form, they shuffle the genes that make the receptors on their surface. The cells use the altered genes to build new receptors with shapes not found in any other immune cell. This shuffling can produce hundreds of billions of different shapes so that each new B or T cell is distinct as a human face. Because they are so diverse, B and T cells can grab a huge range of molecules, including the ones on the surface of invaders. And it says foreign molecules that trigger an immune response are called antigens. First, though, the cells have to get a proper introduction to the antigens. This job is accomplished by macrophages and other immune cells. As they engulf bacteria or their cast-off fragments, the immune cells cut them up into little pieces. They then bring these antigens to their surface, displaying them in a special cup, the major histocompatibility complex, or MHC for short. Parading these conquests, the immune cells travel to the lymph nodes. There they bump into T cells. If a T cell has the right kind of receptor, it can lock onto the antigens displayed by a macrophage. As soon as it recognizes the antigen, the T cells start multiplying quickly into a battalion of identical cells, all equipped with the same receptor. These T cells can take one of three forms, each of which kills the invaders in a different style. Sometimes they become killer T cells, which search the bodies for cells that have been invaded by pathogens. They recognize infected cells, thanks again to MHC. Like macrophages, most cells in the human body can display antigens on MHC receptors of their own. If the killer T cell recognizes these signs of trouble, it commands an infected cell to commit suicide. The parasite within dies along with it. In other cases,
everyone.
activated...
Sorry.
Hello! Okay. Welcome. So in other cases, activated T cells set out to coordinate other immune cells to do a better job of killing. Sometimes they help by becoming inflammatory T cells. These cells crawl their way to the macrophages that are struggling to fight against the rising tide of bacteria. They lock onto the antigen displayed on the macrophage's MHC. That locking acts like a trigger, turning the macrophage into a more violent killer, spraying more poisons. At the same time, the inflammatory T cells help make the cut swell far more than the macrophages can manage on their own. The inflammatory T cells also kill off tired old macrophages and spur the production of new ones to devour their elder cousins. They're like battle-hungry generals. They're good to have around in a war, but can't be allowed to get out of control. Too much inflammation too many poisons created by macrophages, and the immune system will start destroying the body itself. In the third form the T cells take, they help B cells make antibodies. B cells have the same diversity of surface molecules as T cells, so they also have the potential to snag onto billions of different kinds of antigens. After a B cell has latched onto a fragment, a helper T cell may come along and hook onto it at the same time. In these unions, the T cell can give the B cell signals to start making antibodies. Antibodies are kind of a free-floating version of a B cell receptor, also available to clasp onto an antigen from an invader. Once they're activated, B cells spew antibodies out into the body, and depending on the particular antibody, they can fight the infection in several ways. They can cluster around a toxin made by bacteria and neutralize it. They can help the complement molecules trying to drill into the bacteria to make bigger holes. They can latch onto the bacteria and foul up the chemistry they use to invade the body cell. They can tag bacteria to make them a clearer target for macrophages. As the majority of B and T cells go about eradicating the bacteria from the cut, a few sit out the attack. These are known as memory cells. It is their job to preserve a record of the invader for many years after the infection. If the same kind of invader should get in the body again, the memory cells can switch back on and orchestrate a swift, overwhelming assault. These cells are the secrets to vaccines. Even if immune cells are exposed only to an antigen, they can produce memory cells. Because a vaccine contains only a molecule and not a living organism, it doesn't make a person sick, but it can still prime the immune system to wipe out the pathogen if it ever meets up with it again. T cells, B cells, macrophages, complement molecules, antibodies, and all the other parts of the immune system form a tight net that perpetually sweeps our bodies clean.
That was interesting.
To me, it's like a
the Shakespeare immunological text.
Yeah, it's like you're watching this war go on. You're listening to it, and of course it then goes on to.
You make immunobiology sound like swashbuckling.
Well, it sounds like that to me when I read it, and of course it then goes on to say some pathogens escape all of this, but we already know that. But it's kind of an exciting thing to me when I read that. Other people may be bored by that, but I've read that passage again and again and again, and it's just super cool the way it all of this attack happens. All of it's orchestrated and marvelous. So Mel, welcome.
Hello, can you hear me?
Yes.
I haven't done this on my phone. I'm such a techie noob with all this Google Hangout stuff.
I know the feeling.
T Plus, and I'm like, wow, this is a thing.
Yeah, see, I'm the exact opposite. I have my Google Hangouts disabled on my phone, so I don't have to worry about it.
just be careful, Mel, because there's studies that show that the distraction equivalent of using a phone hands-free is the equivalent of twice over the drink drive limit. So, I would hate for you to be involved in a collision because you weren't paying attention. So make sure driving is the most important part of what you're doing here.
Driving is the most important part. I've had a very, very busy day, and I'm sorry I couldn't pop on sooner. I haven't been at home from work yet. I was up to my elbows in HIV antigens, so this is a fun talk for me. It's recombinant. It's just the antigens. I'm not posed as a threat at all when it comes. I'm not like playing with active viruses or anything. So.
Right. Well, Mel, why don't you go into a little bit about explaining what antigens actually are?
Explaining what?
What antigens actually are.
Oh, okay. Antigens. All right. So antigens are essentially on everything that has a protein signature. Antigens are the things that make us sneeze if we have allergies. Antigens are the things that are expressed on plants that our body freak out over when we have allergies. We're like, oh my gosh, it's pollen season. Well, those proteins associated with the pollen essentially are setting off an immune response. So antigens is pretty much, it's a blanket term, but anything that can set off an immune response pretty much in your body. So some people, that's food. food has antigens on it, and sometimes, you know, it can make your mouth swell depending on if you're allergic to it or not. But it's also on bacteria, it's on viruses. That's when you were talking about vaccines. I actually did a video about how vaccines are made and the different types of vaccines. But what you get when you're getting a vaccine is a deactivated version of a virus, but it has those antigens that your immune system would recognize in order to make antibodies. So we have antibodies. Those are the things that attach to the antigens. So it's kind of like a puzzle piece or a lock and key type of situation, to where they kind of fit together really, really well. And I'm trying to find easy ways to explain this. And some of you are like, well, this is probably, you know, this sounds entirely too easy, but that's the best way I can explain it.
Well, funnily enough, we had a couple of videos that showed exactly what you're describing there.
Mm-hmm.
Oh, okay, cool. So essentially, like the job I have, I work in recombinant type of stuff. And recombinants, what we do is we can take E. coli or we can take yeast and we can get it to express a particular antigen, because guess what? The genetic code is universal. So we can genetically alter yeast or E. coli to turn on a particular gene that's associated with making that antigen, and it'll express it. So we don't necessarily even have to have the viruses or, you know, the bacteria or any of that in order to get an antigen to express in an organism. So a lot of what I do is harvesting and purifying these antigens for use in hospitals, research, all kinds of stuff. It just so happens right now I'm working with HIV2 GP36. That GP36 is the DNA coding for that particular antigen, which is used to determine whether or not a person is infected with HIV2.
So this method you're talking about, this is how, if someone is really sick from some kind of pathogen or whatever, that, you know, this is the stuff that they'll be injected with to help them fight this.
No, the antigens that I'm talking about are used for detection and are also used for research in order to make tests.
Oh, right. Okay.
Yes. So, you know, if I wanted to, I could take these antigens that I just purified today, and then I could run what we call an ELISA test or a Western blot. And what you do is you attach antibodies to a membrane for HIV2 GP36. All right. You attach the HIV antibodies that a person would have in their blood, because you can develop antibodies for it. But that doesn't necessarily mean that you can fight off the virus, because there's a two-stage there. Like Jade was talking about, the antibodies have to attach to another cell in order for them to be functional. So you can develop antibodies for a thing and not really fight off the virus. And that's kind of what happens with HIV. So you can develop these antibodies. So we take those antibodies, and there's two ways you can test for it. You can either try to test for the antigen in a person, which would be the actual HIV virus, or you can test for their antibodies. So let's say I'm going to test for their antibodies. So I would adhere my antigens to a membrane, and then I would take a blood sample and run it over that membrane and see if anything sticks. But there'll be markers on my antigens that kind of show up either through fluorescence. If you're into nanoparticles, then that would be Raman spec. But more than likely, you'll have a fluorescent signature to where if it'll go off. Does that make sense? To where you can see it in a microscope. If there's an attachment there, it's gonna register a chemical reaction, and you'll be able to see it. Now, the flip side of that is if I wanted to test to see if somebody had the actual virus, I would stick antibodies on a membrane, then take their blood sample, run it over the membrane, and if it sticks, then I know they have a live virus going on. Have I lost anyone? Yes.
I just wanted to jump in and ask a question, if you don't mind. Sam brought up Chinese hamster ovary cells. Are those part of your research in recombinant antigen?
I'm not in research now. I'm in industry now, but I do work on closely in research in regards to making these processes better on a larger scale for production. So I'm in industry science now. But the mouse ovaries, what was the question about mouse ovaries?
Oh, are you experienced with the Chinese hamster ovaries? They're probably the most popular. No,
I haven't worked with Chinese hamster ovaries. I'm primarily working with yeast right now. So we induce antigen expression through yeast. There's a project that I'm working with. I can't talk a lot about it because
That's okay. We can go back and redact this video later.
I can't talk a lot about the methods and stuff because that's intellectual property.
Yes, of course.
But we do work primarily with yeast because it's easily controlled within, like a fermenter, and you can control the mode of induction of a particular gene in order to get the antigen to express. So I work closely with R&D in perfecting these processes in order to make it on a larger scale. And then I'm the one that actually handles all the yeast, all the purification steps. Nobody else packages what I do. It's from beginning to end. It's all in my hands. So I can't mess up so often. So if I mess up, that's a lot of money. So.
Well, it's very cool. We're very lucky to have you join us.
Well, it's fun.
Have you as part of the community.
Yeah, it's a fun job for me. So I do enjoy it quite a bit. And it's new because it's a bit different because I've primarily done cancer research. And that's the publication that I have coming up at Theranostics. It's to do with a breast cancer test that I helped grassroots when I was in graduate school. I worked primarily on the engineering aspect of 3D printing the isolation of the individual wells. We were trying to do a micro device to where we could have a high throughput test that's a bit more sensitive than the current gold standard, where we detect exosomes, not necessarily the cancer cells, but the exosomes that they release when with the more aggressive strains. That's how they metastasize, is by releasing these small little packages that have genetic code. Other cells can uptake these little packages, and then that can start initiating cancerous types of processes, including, as I was putting in the text earlier, starting the production of certain receptors in the cellular membrane, like the LPA2 receptor, which is what I was talking about, because I just happened to hop on here while y'all were still talking. And somebody posted a question about cancer cells tricking the immune system. And I think that's what I was responding to earlier.
Yeah, each cancer type has its own variation on that same type of process that you mentioned.
Yes, with epithelial cancers, that would be like breast cancer, some skin cancers involving like the colon, anything that is aligning or has skin-like type of tissue, that you can see a high expression of the LPA2 receptor in those epithelial cancers, and that is linked to cellular rescue. So it stops cells from undergoing what's called apoptosis, and that's that programmed cell death that that cancer cells are supposed to undergo to prevent it from becoming, just dividing and dividing and dividing and growing and taking over all the resources, crowding out all the healthy cells. But that's what's supposed to happen. But if you have a high expression of that receptor and you have a lot of what's called LPA, that's lysophosphatidic acid, that's all through your body and it does all kinds of different things. But if you have a lot of these receptors, it prevents that cell from undergoing that cell death. So they just stay alive and keep dividing. And so a lot of the computational chemistry research I did was developing a protocol in order to search for antigens based on known antigens to try to figure out, because you can use various databases. And so I came up with a computational type of protocol in order to find antagonists that would be well suited for potential drug development in order to stop those receptors and make chemotherapy more effective. So that's another bit of cancer stuff I've done.
You're basically describing a means of hacking the cancer.
Yes, hacking the cancer through its communication devices.
Yep, exactly.
I just got home.
Good for you.
I'm sorry I couldn't pop on sooner. It's just been a crazy week, you know?
Don't worry about it. We'll be doing another one next week. Don't know the topic, though.
Well, we still could go on with some more about the immune system, especially if we get Ian in here. And I posted a link. I posted a link to Tommy Hall's favorite site that has some really interesting stuff about some wooish stuff. I don't know if anyone wants to tackle anything on there to explain.
Was this natural news?
Yeah.
Oh, I've had people send me stuff like that before, and I just a lot of times on social media they'll be like, "Well, look at this. This is on natural news."
Yes.
I'm like, "What the heck is this?"
Okay.
What are you talking about?
The problem.
Can I read a couple of these, Jade?
What?
There's too much. I mean, I'm just looking at it like there's too much to deal with. Have you seen how many articles there are?
Can I just tell you my favorite right now? Right, it's near the top. It says, watching scary movies could help boost your immune system and promote weight loss.
I saw that. I saw that when you sent that link before.
Okay.
I was like, what the heck is this? I'm going to scream myself skinny. All you got to do is scream really loud. You know?
Yeah.
When we say swear words, swear words burn extra calories.
Yeah, it's not so much that. The reason why you lose weight is because it scares the piss out of you. So you lose weight, but unfortunately you end up with a puddle on the sofa.
Well, there is that.
Yes.
Listen, I was thinking, right, we've been going for over 2 hours. I want to keep this short. I'm not into the big 4 hour, 5 hour hangouts. But if somebody wants to host an after show, feel free. If you do, host it on your own channel, because I'm shattered again. So I won't be hosting an after show. And you on air, off air, whatever. I think Drag was interested in having a chat at some point after we'd finished.
Well, thank you so much for the invitation again, Fiona.
Now, here's the next question. This is a question we ask it every time, and we hate this question. I'm going to ask it anyway. What will we do for the next one?
What will you do for the next one?
Yeah.
Any
topics that would be worth having a look at? I'm planning. I'm hoping tomorrow night. I don't know if it'll happen or not, but I was going to be doing an off-air with a couple of people if they're available about the absolute basics of DNA.
Mm-hmm.
Yeah, the real basics. I was going to do that off-air with them.
Cliff brought up a good point, because he's mentioned this several times, and he's done so much work for us that maybe we should just take up the poor guy's suggestion for a change.
Yeah, okay, I just saw that. Yeah, I just saw that.
We could also bring.
Sorry, we've got lag.
Okay. Yeah, I was going to say we could also bring in the Black Plague.
Yeah, that'd be a good idea.
Yes.
Right, so the next one, we will go for HIV.
HIV, okay, cool.
HIV.
So everyone go home and watch Dallas Buyers Club. Then you'll know everything about HIV.
Yeah. There you go.
Gosh.
Yeah.
I did do a video on it with the history and immunity and how it came to be and what it was. So if anybody wants a crash course.
There you go.
A good idea.
Yes,
listen, it might be an idea if Mel and RSC have a chat about this and work out what they want to cover. You split it between them, and also Ian and Hogtie Champ. And I'd love to hear from Ian about the attempts to get a vaccine.
Well, I can make another little list like I usually do of subjects to cover. And as I said, we want to look at the history, we want to look at the structure, we want to look at how does it attack, but we also look at how it's being re-engineered to fight cancer and other diseases, and also to look at how essentially those of us who are survivors of the people. Of the descendants of the survivors of the Black Plague have a little bit of a leg up in fighting HIV. It's just an interesting little historical tidbit, and I'd be happy to cover that one if no one else wants to. But
okay, cool. So, well, it just remains for me to draw this to a close. So I want to thank the audience for sticking with us through this. I hope they've learned something that they wanted to learn. If they have any questions, anything that they weren't clear on, contact one of us, whichever way you prefer. I want to thank RSC for his presentation. He seems to be really good at these presentations. And I want to thank everybody, nearly everybody, for keeping the live chat pretty civil and for bringing their questions to that. So next time, and the target is next Thursday night, same time, we will do a hangout on HIV. It's history, it's pathology, it's genetics, and the attempts to find a vaccine for it. So with that, I will ask all of my fellow science bitches that are in the room with me to bid everybody good night.
Quick thing, Bullinator's going to have an after show.
No, Drag's going to do it, isn't he?
No, it's that Bullinator starting a hangout right now.
Yep.
Oh no,
for an
Drag
after. I don't know.
Get it straight, because it says Bullinator. I'll have after show, or I will have after show.
No.
So
hide it.
It says Bullinator says I am starting the hangout right now.
Oh, okay, well, whatever.
Bullinator, thing to do is once this goes off, you could stick the link for your after show in the comments.
Right.
Yeah. And Drag said to Bullinator, "Do you want to hang an after? Do you want to host an after show?"
Oh, okay. Yeah.
Yeah. Oh, he's got a watch link.
Yeah.
Okay.
Right. So anyway, could my fellow bitches please bid our audience good night? Say bye, everybody.
Good night, everyone.
Good night.